Welcome to the training session on external quality control for the Hain MTBDRsl with the PPAW regimen. This training session was prepared by the International Tuberculosis Research Center. Before we move on to the detailed explanation of the EQC procedure, let me remind you of the two critical elements for a valid experiment. First, equipment validation is the most important step before starting any experiment. Make sure your equipment is validated within the period stated in the management plan. If the equipment validation is near its expiration date and the study cannot be completed within the initial project period, I strongly recommend that your instruments be validated before starting the project. Second, I would advise you to focus on performing the experiments accurately. The number of samples you can handle in a day and how fast you perform the experiments might not matter at all. Instead, focus on the basics. Pipette slowly and accurately, and change the micropipette tip after each sample. With these two simple but significant factors in mind, let us get onto the topic. It is my understanding that most of the countries participating in the LEAF-TB project have been using the Hain MTBDRsl to identify exterior TB, and several countries have introduced EQC and EQA programs. This material was prepared to provide support for EQC for those participating in the LEAF-TB project, just in case the participating countries or laboratories intend to complete additional EQC of their data. The Hain MTBDRsl should only be used for the evaluation of baseline samples to determine whether a patient matches the inclusion criteria of the LEAF-TB project. If a group wishes to add the ITRC EQC evaluations, it is recommended that these are completed prior to starting the LEAF-TB project. However, you can still perform EQC during the project period to monitor your quality performance. As this presentation is about quality control, I will not describe the detailed procedures of Hain MTBDRsl. You can simply follow the instructions in the user's manual from Hain. Instead, the ITRC will provide a panel of pre-qualified DNAs for EQC, and you may conduct the experiments. ITRC will provide a panel of 10 DNA samples consisting of a positive control, a negative control, and 8 experimental MTBDNA samples. Each experimental MTBDNA sample was selected to represent a different type of resistant determinant and resistant characteristic. Slide 5 and 6 show sample EQC test panel results with the color indicator stripes on slide 5 and the interpretation of the results on the slide 6. Each batch of the EQC DNA panel contains a different set of DNA samples, and you will get one of these batches upon request. In some DNA samples, certain signal bands may appear to be very faint, but this is intentional as it is part of EQC. Samples with weak signals were selected to monitor your performance and decision making. The table on slide 6 presents the sample results from one of these panels and describes how to complete the reporting form. A complete form is presented on slide 7. This form is very similar to the Hain MTBDRsl reporting form and consists of two parts. The upper section is designed to record the presence or absence of specific bands in each target gene or locus, and the lower section, which is highlighted in a light purple color, is used to check the sample for resistance to each test drug. Given this, let me show you how to interpret the results. First, each signal on the stripe is not directly recorded in the result table. Instead, you record your interpretation of the strip that is positive or negative in the table. Second, Any missing wild-type probes are marked with minus in the upper section. Third, if any signal is detected for a mutation probe, this is marked with a plus in the upper section. And finally, drug-resistant characteristics recorded in the lower section of the form are marked with a plus only when they are interpreted as resistant. Susceptible cases are not marked with a minus and instead are left blank to avoid any possible confusion. This slide shows a sample reporting document for the EQC samples. The method recommended by the ITRC for extracting DNA from sputum samples to minimize the risk of an invalid result or failure in the Hain LPA is shown on the slide 8 and 9. Although there are many commercially available methods for extracting DNA from sputum samples, Genoase is one of the most widely used and thus is the one recommended by the ITRC. This is only a recommendation, and if you already have a well-established method in your laboratory and the quality of the extracted DNA is good enough for the routine examination, you do not need to change your method to Genoase. This slide shows the step-by-step procedure for Genoase protocol. Transfer 500 microliter of decontaminated sputum into a 1.5 milliliter screw cap tube. Centrifuge at 10,000 G for 15 minutes in a standard tabletop centrifuge with an aerosol tight rotor. Discard the supernatant and resuspend the pellet in 100 microliter of lysis buffer by vortexing. Incubate the samples for 5 minutes at 95 degrees Celsius in a water bath, and then briefly spin down. Add 100 microliter of neutralization buffer and vortex the samples for 5 seconds. Then, spin down this mixture for 5 minutes at full speed in a tabletop centrifuge with an aerosol tight rotor and use 5 microliter of the supernatant for PCR. This brings us to the end of our training session on external quality control for the Hain MTBDRsl. We hope that you have enjoyed this session and found this material useful. If you have any questions or inquiries, please contact the ITRC at mlab.itrc@gmail.com. Thank you very much.